So my title of the talk is 3D bioprinting cardiovascular tissue model for space-based applications. So a brief overview, some background, what is the motivation, why were you looking at space-based applications? And then we're going to talk about 3D bioprinting. You heard a little bit, actually, it may be a good sort of segue to the previous talk by Dr. Webster, who sort of talked a little bit about 4D printing. So we'll be talking about some bioprinting methods here. And then we'll talk about the characterization, how you have done it, some results and conclusion. And of course, this is a wide open field, still a lot of ongoing research. So I'm going to give some perspective, of the research we are continuing in this field. So if you are not very familiar with the 3D bioprinting, it'll be as good a perspective what it entails. So you can basically print multi-material along with multi-different types of cells to create a complex tissue structure. So and the biggest advantage is that you can control the tissue structure by using your printer heads. How do you want to structure it? How do you want to deposit the cells? What density? So in essence, you have a complete control to create a 3D tissue structure. And we can use different types of biomaterials, which will simulate the different type of tissues characteristics. Like we all know that the extracellular matrix of different tissues varies. So based on the different type of biomaterials and different types of cells combination, you can create that architecture quite convincingly compared to maybe in a traditional 2D culture. So the word bioink, if you are not familiar, is a hydrogel material which is used to encapsulate the cells so that you can provide the supportive structure to the extracellular matrix. And important characteristics is that it's also going to protect the cells from stress during printing. Because one of the important things is that when you do bioprint, it's a common question, are the cells going to be stressed? Of course, but as long as you can control all the printing parameters and the type of biomaterials, which is the supportive environment, the cells stay well alive. People have actually kept it for 6 months. And the other important key, how do you keep the cells alive? The next slide, the bullet, vascularization. It is the key because you can print and do some media exchange for a day or so. It will stay healthy, but we want to keep them alive, especially for space applications. When they go for missions, you need to keep them, sort of live and healthy for extended period of time. So vascularization is going to be the key. How do you sort of address the viability of the tissues? So the development of vascular network within this intricate tissue structure is going to be critical, as they're going to provide the transport of nutrients and of course the removal of waste products, which are critical to maintain the high cellular viability over an extended period of time. So the key takeaway will be that you want to maintain a vascularized tissue for an extended period of time, and the goal will be to develop autonomous processes, which is still a challenge, but that's sort of everybody's trying to hit that goal. So again, give a little bit of perspective what it entails. Like the key terms here in 3D bioprinting will be extracellular matrix, which will be your biomaterials, which you'll be selecting for the right type of tissue. Then you have to mix the cells, appropriate cells, and then you have to do the vascularization. So these are the 3 key broad terms which will entails in a 3D bioprinting of tissues or organs. So I mentioned that bioprinting maybe sound exciting, but we have to be really very careful so that cells are sensitive, that we don't sort of damage the cells. So how do you sort of look into it? So one important thing will be there are different types here. I made it based on extrusion printer, but we actually have also light-based processing printer. We also have volumetric printing, but most people are familiar with the extrusion-based printing. And in extrusion-based printing, your pneumatic force pressure or the mechanical force, which is applied during the printing process, has to be controlled properly. And also you have to look at the different types of nozzle diameters and the extrusion rate, because other important part is the shear stress, what is experienced by the cells during printing. You have to control that. And so you really have to look at the viscosity of the bioinks. And we know that the bioinks have different viscosity, and that is going to dictate how much pressure what temperature you have to control to print the right combination of biomaterials and cells to print the mostly viable tissue. So it's really a complex interplay between shear stress, viscosity, and other rheological materials so that you can maintain, and these are the basic, that are much more involved, but in a higher level, these are the important parameters which you need to control during, to maintain the viability of the cells during the printing process. So that brings us to the most exciting words. I'm sure you must have heard of the word organ on a chip, tissue on a chip. These are the powerful models to study disease mechanisms and improve treatments. And because doing animal studies is great, there are always advantages, but there are, as I said, the focus is on space. And I know that NASA is really interested in tissue chips, organ on chips, because if you're going to go to a Mars mission on the moon, these are long duration missions, and they want to develop an autonomous organ on a chip system to maintain the physiological conditions and experiments. Doing animal experiments is expensive. Even FDA, if you have followed the news, have recommended minimal use of animal studies. So that's the reason this organ on a chip, tissue on a chips are really getting wide emphasis right now as a real model to mimic human disease and developing countermeasures. Actually, detection is important part, but the real key for any disease at the end of the day, we all know, is to have the right countermeasure, because we can detect everything, but if we don't really develop a countermeasure, yes, I mean, we need that, but we need to be able to know how can we do that to prevent that. And the space environment, the microgravity is a very key, because it's going to provide you an environment where you can look at a lot of disease models much easily. Like one of the areas of research right now is aging research. You know, I mean, even you heard a little bit on the previous slide to Alzheimer's and stuff. So aging is a major problem right now and it has been shown, like the astronauts, when they go to for 6 months of mission. In fact, I was in a workshop a couple of days ago, and I've read the data, and they showed that, you know, an astronaut's body comes back as a 6 months gives you a 50 years aging body. Just think of it. Do they come back? Of course, they recover to the previous state, and they're still trying to understand what are the mechanisms and stuff involved in that. So aging, for example, is a major emphasis right now for disease modeling, and the microgravity environment is going to give you the right platform to do that. And how do you do that in a microgravity environment? Organ on a chip, tissue on a chip, because they will let you emulate the intrinsic molecular profiles, mechanics, and function of the vessels. And again, they emphasize that you can really control your environment of the tissue structure. You can apply the right biomechanical stimuli in both duration and intensity, and that makes it an ideal platform for, I mean, I just put infectious, but again, you can do for any type of disease modeling and treatment. So it's sort of a real platform which can be used not only for space, also for art-based applications. In fact, right now, if the NIH, for example, is having solicitations for developing tissue chips for space. I mean, because the value of space is so significant in terms of identifying certain type of disease models, that is providing like a platform that one will be using, and the tissue chip is the right environment to do a lot of these studies. So again, my focus here today on the cardiovascular type of tissue. So we know that normal endothelial function is key, because that helps you to maintain the balance in mechanisms between contraction and relaxation process. So if you have a damaged endothelium, then you're going to have a vascular dysfunction, and which means to vasoconstriction and cardiovascular disease. So there are various markers you can look into it. A couple of the markers we are looking at, one of them will be first the nitric oxide. So we all know that nitric oxide plays a cardioprotective role in our as a signal messenger in our cardiovascular system, and it again helps in against the onset and protection of CVDs. So in healthy conditions, nitric oxide is the major endothelium-dependent vasodilator which is found in our body. But if you are, I mean, maybe I'll not go to the very details of the biology, but the angiotensin-renin system is the key that sort of comes into play. So the conversion of angiotensin by renin results in a fall in a blood pressure, and that will result in the production of angiotensin 1 that leads to angiotensin 2, and via the angiotensin converting enzyme S, and that is going to result in increase in blood pressure, and that turns off the initial stimulus, and you can get cardiovascular disease. And then, of course, people are looking into the different blockers of the angiotensin 1 and 2, both blockers. You can see here, these are the ones you want to develop, these receptor blockers that can prevent the onset of cardiovascular disease. So the angiotensin-renin system, but from our perspective, I just want to focus the word nitric oxide because we are looking at the mechanisms which causes the cardiovascular disease. And the other is the reactive oxygen species, because these are the two key parameters we believe, at least from our perspective, are very important that contributes to the vascular disease and stress and oxidative stress. So when you're in vascular disease, your ROS level is going to increase and your nitric oxide level decreases. So you really need to maintain the right balance between this production of these radicals and that a body is able to counteract that properly. So these are the two important parameters that you will be measuring during this process. And as I also showed you in the previous slide, that angiotensin 2 again is the key parameter, one of the which leads to the development of a CVD. So in a vascular disease, again, ROS production increases and nitric oxide decreases, and especially the decreased production of nitric oxide in many pathological conditions due to the response of increase in angio2 upregulation will have serious problems in endothelial function and vascular functions, which I sort of again want to emphasize that why we are looking into a couple of these parameters. So very high level objective. So we have to develop 3D bioprinting techniques so that we can develop vascularized human relevant phantoms, tissue mimics, what you can call. And then you also demonstrate that the Y organ chip is an ideal platform for disease modeling and treatment. And then we want to sort of maintain the viability. Right now, we are focusing on 15-30 days. I know for the longer duration, we have to look at 60 days, 90 days for missions. But right now, we are targeting those missions, which will be more like 15 days, because we have to make sure that at least we can do that level convincingly. Or we are actually more of 30 days, but I still have some of the papers are still ongoing and publications and under review process. So far, we have sort of shown easily that 15 days, and then we do a lot of characterization and perform measurements of these bioprinted tissues that can maintain the functionality of these tissue structures. So if you're familiar with the bioprinter, you may have seen this is an extrusion bioprinter. It has got 6 heads. As I said, this gives you, we are not using 6 heads, but ideally you can use 6 types of materials and 6 types of cells. Complex, we are using 3 at most. So, I mean, as I said, there's a room. It's not just a regular printer where you press a button and you get a tissue print. Lot of optimization, pressure. Just yesterday, in fact, we are doing some experiments in the lab. We are doing one type of new biomaterials, and as soon as we are mixing with cells, the extrusion parameters are changing compared to just without adding the cells. So it is that level of simply complex to think of it. So these characteristics has to be really well controlled, and that's why you could see that the print heads beds can range from this high. I mean, you definitely never go to 60 degrees, but it has the potential. But typically, you like with our controls, we have anywhere from 10 degrees to 25, 30 degrees based on what type of biomaterials and stuff we are using. So it's a lot of controls and optimization process and the pressures that involves, in this the 6-head bioprinting process. So again, as I said, what are the typical in a bioprint is cell mixing, bioprinting, and the other important is cross-linking. What happens when you print? These are gel materials, right? So you have to make sure that you cross-link it, because by the time you print a layer and then it goes to the next to print the next layer, that supporting layer we may already fail. So how do you prevent that? By cross-linking. And you can do different types of cross-linking. This printer came with a UV light cross-linking. So after, again, that's another optimization process. You can cross-link after every layer for 5 seconds, 10. So each step in a bioprinting can be a project by itself. It's that level of, I'm saying it looks at complexity, but once you know how to do it, then it's not that difficult. But the starting point of a bioprinting process is a lot of optimization process. And I want to acknowledge that. I don't want to give you a feel to the listeners that it is a press where you press a button, you come out with a new tissue. It's not that. It is, but it gives you nice structures, but you go to the optimization process. So very typical, I'm sure you have seen that you use a lot of CAD models, use slicers, and then you You can do a lot of customization, the G code, MATLAB. I mean, this is just the way we did it, but there are many, many programs. In fact, we are doing a little bit different way right now. This is maybe 6 months old technique I'm staying. We're actually using some new slicers and stuff, which came with actually the printer. The printer actually added a software that does a little bit better sizing. So, as I said again, the bioink will be an important parameter, and optimization, we're actually doing some mathematical models because the nozzle diameter, the pressure, the printing speed, all these are very important to be controlled during the bioprinting process. So, of course, it's going to depend on the viscosity, dispensing type, the type of CAD geometry you're creating, the shear stress, sort of all this. And so we're doing actually mathematical modeling, which I'm not showing in this one, but we have done a lot of modeling. This shows you just a snapshot of the parameters involved with our extrusion printer. You know, the cell densities involved, the needle diameter, the print speed, the print head temperature. In fact, I told you, I said 22. Actually, yesterday we were working with the materials where actually you have to print at 14 degrees. So we have to see how viable is that. So you may want to control these, but sometimes the type of biomaterials and the cells that can have some impact. And then the print bed temperature, the pressure, infill density, the resolution, cross-linking. Again, we're doing after each layer, how long? That's another optimization process. The wavelength, I mean, it is UV, but again, there is some flexibility in the range, and the cross-linking distance to the center of the build. So these are just some snapshot of some parameter. There is much more parameters when involved in a printing process. So again, you have a G code, and it gives you all the translation rates, extrusion, and so these are all sort of and then the 6 heads, whatever the printing sort of a software entails in that. So there are many ways, as I said, the key for bioprinting will be to print vascularized tissue. So there are many ways we are looking into it. One way we actually started is sacrificial bioprinting. And as the name says, you can see in the picture what it does. You actually first create a sheet of material in your tissue chip system where you're printing, and then you print your other matrix of tissue over that initial sheet, and then this initial sheet is made of some sacrificial materials, and then you sacrifice this out. So what will be left with will be some vascular channels. So as I said, we started this. We're actually looking into some other methods, which I'm not presenting today in the details, but this is one of the very classical methods. We started with the classical method, sacrificial bioprinting, to create vascularized structures. So it's because again, key I want to emphasize is vascularization. I mean, you're going to see a lot of papers able to print, but are they able to maintain the perfusion for an extended period of time? That's going to be a key. And other important thing is you want to make sure that it is going to be whatever you're printing is just not very specific. You're going to type various, like a brain tissue we're working on. I'll show you that at the end. And cell types, different types of extracellular matrix. So different type of combinations. It's sort of a permutation combination. What may give you the right structure? So I'm going to focus next on the few, because the focus is on the space, on microgravity environment. What we did in a simulated microgravity environment. We are still waiting for some flight experiments, but we thought that the key is to printing on the microgravity simulated environment. And so these are some extrusion printers. We also have a digital light processing. See that we have actually printed much more advanced, structures, but those are papers under review process. So these are some of our initial vascular structures we have printed using a light processing printer. We're also using a volumetric printer. So this is one design of our tissue chip system. As you could see, it has a perfusion base, so you can maintain them over an extended period of time. And I told you about sacrificial material. Pruronics is a very, very classical. I mean, as I said, this one we didn't invented it. This is a very classical materials we have been using as sacrificial materials. We are using some Gelma-based biomaterials, but a lot of combinations. We actually have been working with the company. We have an NDA with them. They make different types of biomaterials, different combinations of bovine, or just along with the details, mixtures that, and we are seeing that you get so much variety in terms of structure, functionality variation. So we are really working with them very closely to develop a platform or like a database, what type of materials, because you can, in fact, initially when we were started, we actually were buying it from a very well-known company, Advanced Biometrics. That's one of the biggest company in US who supplies a lot of biomaterials. But we realized that we are losing control. And then I sort of took a breath, for a month or so, and then developed this relationship. And then we are getting really, so you're not seeing these results here because part of this NDA, and then part of it is papers under review. But even with these, we got very good prints. But now we have even a higher level of prints, well optimized for different materials, different types of cells. So again, I want to emphasize that the selection of the biomaterials is going to be very key for developing the right tissue structure for biomedical applications. So going to the characterization, obviously you have to do rheological studies because you're looking at viscosity of these materials. You have to look at the viability. Obviously, there's a key parameter. We also looked into some MTT assays, and then we looked at PCR, and then obviously our biggest emphasis is oxidative stress, because I'll show you the data what we got using a simulated microgravity and also some morphological analysis. Just some sample, I'm showing it to you. So the important thing is that you want to be able to measure the viscosity as a function of shear rate. So that's you really want to characterize that, because you want to see what type of bio ink you will be selecting for your printing process. So this shows you how the viscosity and the shear rate. So you could see that for the lower viscosity, your shear rate will be lower. So that gives you, so that means you can, and one of the advantages, and these are shear thinning materials, that in microgravity, as you know, that you can actually print with less bio ink. So people may ask that why you want to use the International Space Station for printing, because it provides a natural environment to print with less viscous bio ink. And so that's why you don't really have to worry about the viscosity of the ink. Because of lack of gravity, you're able to print with less viscous, and that actually provides a great platform. And that's the reason why ISS is an important platform for printing. And we also did some rheological analysis where we look at shear storage modulus, versus cross-linking time. As I said, there are many parameters. We just showed you one here again, just to show how can you optimize this bioprinted process. So we are printing some initially simple vascular channels, and we want to make sure that we're able to maintain the viability, because these channels were seeded with HUVEC cells, and even after 16 days, we are able to maintain 95, 98% of cell viability. We use a standard live dead assay for assay kit, and for our viability studies, the calcium stains the green, and the EDM homodimer is a red, and you could clearly see barely, maybe even here, one or two. So also you have to also look at the flow velocities of our optimization of a microfluidic system. So you could see that that is another important parameter which can be also controlled. So even at this rate, we got a liquid of red cells, but here in these two parameters. So we are able to be convinced that all the parameters are optimized for our applications to get high cellular viability. We also did some quick MTS just to make sure that's another way to look at the viable cells, but we more like the live dead assay. And then, of course, bioprinting is great, but we want to make sure that you can get a good RNA, because our goal is to look at some microRNA biomarkers for disease modeling. Because for cardiovascular disease, we are going to show that there are certain types of microRNA which are either overexpressed or underexpressed. So we were able to extract the RNA, and initially what we did, we just before doing the microgravity experiment, we want to make sure that the tissue, because there are always skeptics, can the 3D bioprinted tissue sort of replicate the real science? And we wanted to show them that all we did our bioprinted tissue, and then we co-cultured them with the young and old HUVEC cells printed, because young, old means their viability will be of course less, and young means young, as the name suggests. So we sort of co-cultured them, and then we sort of did the cDNA and the qPCR, and we wanted to look at the expression of the microRNA just to demonstrate that these 3D bioprinted structures is going to replicate what you're going to see with using an animal model, because it's a very classical approach. People will extract the RNA and do the PCRs to show, which type of microRNA is extracted and how they're expressed. So again, here is our and what we did in order to generate the older HUVECs, because one of the approach we're looking at aging, and aging causes some senescence. So we actually irradiated them with X-rays, so to create these older HUVECs, and then we co-cultured them with a non-radiated, irradiated. So using the radiation in this case, a control before our microgravity experiment, like a control to look at the expression of this microRNA for disease modeling. and we got able to get a good RNA yield, as you could see from this data, young, old. And then what you saw is interesting, which you wanted to see, that this microRNA expression with older was increased, which is expected, because these are the results people have shown in animal studies, that these pro-senescence microRNA showed a 2.2-fold increase in expression, and this particular microRNA 217 showed a decrease in the expression when cultured with old HUVEX and versus young HUVEX. When I mean by old, it means irradiated. Actually, I want to be very, very specific, and suggesting that the cells are influenced by cell cultures externally. That means the bioprinted tissue is playing a role. It's not an artifact, because a lot of time people may say, oh, how do you know this expression is correct? So these expressions have been manifested in animals, and now able to demonstrate that very significantly just through a sample result. We did extensive studies to show that how the bioprinted tissue can be the right platform for these studies, because we want to make sure that you can replicate this environment, these data, when you use microgravity environment, which is supposed to cause stress. As you all know, the microgravity environment, that's the reason NASA is really interested in these ISS studies. And I'll not go into the details. So we wanted to do the nitric oxide assay. I told you how nitric oxide is one of the vasodilation mechanisms in our body. So what we actually, again, we want to make sure that there are no skeptics in our work. So we used an agent to decrease and increase the expression of nitric oxide using this bioprinted tissue in the lab. So we used LNAME, which is an inhibitor, to see if nitric oxide will contribute to the proliferations of the cells, and then use sodium nitroprusside, which is able to enhance it. So we want to really make sure that the mechanisms we wanted to study can be replicated, which will be done in a real environment, can be done in lab studies. So we did the whole assays in a lab settings to show that how we are able to increase the enhance this, with the control and sodium nitroprusside, and then if you use a LNAME, it brings rank down. So sodium nitroprusside basically increases, as you could see from the color, the expression, and then if you use LNAME, it brings it down. So we want to really simulate, if you go to microgravity, what you're going to see with this bioprinted will be a true representation of the mechanisms. And that's why we did these lab experiments, just to demonstrate the importance of it. So you could see that what we expected, sodium nitroprusside is an enhancer, it's going to increase it. And then if you use LNAME, it brings it back. So we are very satisfied that all these mechanisms work very well. And then we are using the ROS. We did very similar studies in the lab setting. These are the assays, very typical. Again, we are using standard assays available from Thermo Fisher Scientific for the NO and the ROS mechanisms. We also look at the morphology, because it's very important to look at the networks, because the important thing in a bioprinting, you want to look at the networks of the channels, how they're sort of interconnected, because for vascularization and providing the nutrients. So you want to make sure that we are able to put some fibrinogen, and that helps to increase the vascularization network and also provides more increased structural rigidity. So we did all of these sort of studies just to make sure that we understand that the bioprinted tissue structures sort of replicate the structure, the true environment of a native tissue. And then we also did some myography studies. So we want to look at the myography to look at electrical and neurological activation, to look at the muscle functions. And so we looked at the force and production and fatigue, and because that's very important for cardiac tissue. So we used a stimulation system to look into that. So myography will allow us to test any type of configurations for contraction and relaxation properties under the field stimulation system for various factors, which was very critical. And then I'm going to give you a snapshot. These are a lot of data under review at Kennedy Space Center. This is the microgravity simulator. So this is basically like a rotating system where you put your samples and you put your controls. This is inside an incubator, and it rotates. And there's a software you can create. I mean, we did like zero. I mean, microgravity is 0, but you can control, like if you go to moon, it's not 0, it's like 1.6. So you can create any type of environment you want to create and they're going to be rotate. So we're going to keep the samples for that amount of time. So these are some of your printed structures. It's going to be not very clear here, sort of floating here. You have to make sure when you put this in a microgravity simulator, there'll be no bubbles. That was a challenge. So you have to find, in fact, we will be repeating this experiment in about 4 weeks. And we're trying to see that what type of better systems we can use to hold these tissues, because make sure that way, because bubbles is going to play, as you know, in a simulated, if you have a bubbles, that's going to impact your results. So a lot of learning lessons. We have done this a couple of times. We learned a lot of lessons, and you're going to repeat that and sort of be ready. So with our flight experiments, so this is showing you one data. There are a lot of optimization. I mean, I'm showing you one of the older data compared to a control. You could see that this is with the margin. We didn't see much of oxidative stress increase, but for 72, we saw much significant increase. And we did this for a whole number of samples. So this was based on maybe 4 or 5 samples. We've actually processed after that 5 or 6 more samples, and you see more convincing, because part of it also depends on the bioprinting process. So we have some samples, we tested different types of materials. So different types. So we saw it's like a matrix, and at the end of the day, we have to sort of make sure that whatever materials will be the right one to use for microgravity environments. So it's a lot of optimization process. So in a sense, what we really want to emphasize that real appropriate bioing is very critical, and mixing with the cells for the development of your structure, and the viscosity of the bioing is important. You need to have a good real-time. One of the applications we are looking at space. As you know, there are a lot of space flight implementation partners. Once we go into space, they have microscope-based systems where they can monitor the cells, because it has to be autonomous process. So one of the things is that how can you develop an autonomous process to sort of monitor all of these things, because astronaut time is going to be very key, busy, and some of these initially maybe, you know, so we have to really make sure that more autonomous. That's the reason the tissue chip autonomous is sort of the key buzzwords. NASA is like sort of looking. We are slowly trying to work with iPSCs we have in our lab. We have been just playing with it. We have printed, but again, I'm not showing anything, because that's sort of, we have to really go. As you know, that induced fluorescence stem cells are the ones which is the direction for personalized medicine, because you can get cells from your skin, and you can produce iPSC cells of any organs you want, cardiovascular, brain. So that is the key for personalized medicine, and that's the one actually we'll be doing it for our tissue chip for flight experiments. So we are actually looking at iPSCs right now in our lab. We're also actually going to work at neural brain model, because that's going to be used for aging. So we are looking at neural cells and bioprinting with specific neural cells. We want to add more complex astrocytes and other, but we just have the neurons. It's a complex process optimization. Then at the end of the goal, as I said, I mean, we have collaborators, of course. Not you can do everything yourselves. End of the day, you want to have a sensing system that can provide all the stimuli, all the sensors for an autonomous microfluidic chip on a system. And so for when you go to space, microgravity is one important aspect, and the other important is radiation, for which NASA is really very worried, because for for ISS, radiation, you can have the right environment. You can sort of, when you don't have a solar flux, you can sort of control some of your EV activities, extravehicular activities during the spacewalk. But when you're going for longer duration, like a further mission like Moon and Mars, your radiation is going to be a key factor. And that's the reason we are trying to get some experiments done. NASA has a Brookhaven National Lab, a radiation facility, which simulates a lot of variety of ionizing radiation which exists in the space. It's like a competitive process and very demanding to get into that to get the data, because they're also open for a short amount of time. It uses a humongous amount of energy, so they only operate within certain amount of period of time. And so I'm starting to, that's next in my queue, some proposals pending to take data over there. So whole idea, for example, the cardiovascular disease, we know that the oxidative stress, your NO will be decreased, your ROS will increase, and then we want to look at the mechanisms and then use some angiotensin receptors blockers to reduce the oxidative stress. So that's sort of the overall ideas. So as I said, we have done up to 30 days right now. We are looking into the tissue structure, vascularization. How can you really maintain these tissues over a period of 90 days? Because that sort of is going to be the key. And we are looking, as I said, the response to angiotensin 2 receptors, and then we want to block them with some drug with losartan, and then we are looking at the combined effects of microgravity and radiation, which is very hard to do, what I understand, at Brookhaven National Lab. In addition to the radiation, they're also building a chamber which can also simulate the microgravity. So that will be the real sort of right environment where you can look at the combined effect, which is going to be the key. And as I and related treatment, since I talk of cardiovascular application also in space, but it's also application for Earth. When people do chemotherapy, like doxorubicin, it's a great chemotherapy drug. But we all know, if you read the literature, when people get treated with chemotherapy, they get cured of cancer, but actually they die of cardiovascular disease. The cardiotoxicity after cancer treatment is actually a more cause of death, if you follow the literature, versus the cancer itself. I mean, people may not believe that, but that's true data, and you can Google that to find the data. So we are looking at that application. That's the reason, agencies like NIH and is also interested. They were, even NSF, National Science Foundation, they never thought of space, but we sort of are now part of initiative also of NSF to develop a center for space research with the consortium for universities. So we can look into all of these mechanisms, because it's going to help you to benefit the life on Earth. So I want to acknowledge, some of that. I mean, for this part of the project, you have much more funding source, and National Science Foundation, Florida Space Grant, Cytwing Solutions, and PS Instrument for supporting this work. Thank you. And if you have any questions, I'd be happy to take. I guess we have one or 2 minutes left.
Thank you very much, Dr. Kunal Mitra, for sharing the views on the topic 3D bioprinted cardiovascular tissue model for space-based application. If there are any questions, the floor is open.
Okay, so thank you again for listening. I'm not sure, it's good afternoon, good morning everywhere, maybe good evening. So everybody have a nice rest of the day. Thank you.